time for TWIV, this week in Virology. This is episode 392, and today is May 31st, 2016. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code, what else? Microbe. We're coming to you today from the headquarters of the American Society for Microbiology, which is in Washington, D.C. We're on the eve of a special meeting on Zika virus, which is called What Does the Biology of Flaviviruses Tell Us About Zika Virus? And this is a jointly sponsored meeting between the American Society for Microbiology, ASM, and the American Society for Virology, ASV. This is an unprecedented joint venture, hopefully a sign of more good things to come in the future. And tomorrow, we're going to have a full day of Zika virus. We're going to have flavivirus experts talking about their work, and I have been privileged to be able to take 4 of them and do a TWIV with them tonight and give you a flavor of what's going to be going on tomorrow, because not everyone can come to the meeting. So let me introduce my awesome panel of virologists, starting there on my left. He is from Washington University in St. Louis. Michael Diamond, welcome to TWIV.
Thank you, Vincent.
And on my left, from the University of Chicago, Michaela Gack. Welcome. Welcome to the podcast.
Great to be here.
Pleasure. On my right, from the New York State Department of Health and also SUNY Albany, Laura Kramer. Welcome.
Thank you.
And finally, all the way on the right, someone I've known for many, many years, maybe as old as I am, right? He is from my town, New York City, the Rockefeller University, Charlie Rice.
Great to be here.
I finally got you on TWIV. I invited Charlie. He said, yeah, maybe I'll come. I said, no, not maybe. It's either yes
I came.
or no. You came.
You're here.
I want to start off by, so we're going to have a little chat today, folks. And, this is TWIV. I don't have my usual co-host, so you're going to be the co-hosts of TWIV. And so, on TWIV, we talk about virology in a conversational way. So, although I'll ask you some questions, I do want you all to chip in and chime in. And I know some of you will want to say things all the time, and please do that. We have an audience here. Thank you for coming. If you have questions, there's a mic there that you can use. We have an online audience. There's a Twitter feed where you can put your questions, and we'll answer them. There's also a chat on YouTube, because this is being live streamed to YouTube as well. So let's start out. You are in an elevator with a billionaire, and you have 30 seconds to convince said billionaire that your work should deserve a hefty check. 30 seconds. Michael, what do you tell them? What are you doing?
I think what I would tell them is we're trying to develop animal models of Zika pathogenesis to answer fundamental questions about how the virus causes disease. And really in a couple of areas. One, how does the virus lead to in utero transmission and affect the fetus? What are the areas that it affects? Why does it cause particular clinical manifestations that we've seen that seem to be unusual? The second one that's really puzzling to us is, why is the virus persistent in some areas of the body that maybe related viruses have not historically had that? So this virus causes sexual transmission, although it's probably not the major transmission. Mosquito transmission is, but still it's very concerning. Did I lose my money already?
No, you're at 30 seconds.
Okay.
So that's good. We'll come back to you.
All right.
Michaela.
Well, I will explain what my lab is doing. So we are really trying to understand in mechanistic detail how Zika virus and other flaviviruses suppress our immune system. So most viruses, they suppress our immune system, the innate immune system, the very early one, and the adaptive immune system. And we are trying to identify novel host-virus interactions, and these could be targeted eventually in the future for development of novel antivirals or for the development of live attenuated vaccines or other kinds of vaccines for these viruses.
Laura.
Well, putting my research hat on, we're trying to understand what about the virus, the vector, and the vertebrate and the environment allows this virus to be so explosive in its transmission and spread throughout South America, and what is the risk to other countries, as for example the United States, putting a public health hat on. How can we diagnose this virus better to allow us to facilitate women who are pregnant so that they know whether they're infected or not, facilitate the diagnosis. And putting the two together, what's the public health risk of Zika to the US?
All right, thank you. Charlie.
Well, this is a tough one, 30 seconds. I guess I would say if you want to make the world a better place for yourself, your kids, and your grandkids, you should support your local virologist and virologist. And you know, it's you never really know where the next Zika is coming from, and it's important that we sort of continue to support a broad research base to be ready for when the next unexpected disease appears.
Thank you. Actually, we do have a course here at ASM to help you develop a 30-second elevator pitch. And it is an art that few of us know how to do, although you all did very well. So we have, you can see the range. We have pathogenesis, immunity. We have the mosquito lady here. We have the virologist, the virologist who's done many things. Let me ask you one more thing, all of you. I know you are working on Zika because you're already published 300 papers on it. Well, maybe not 300, but a few. Michaela, are you working on Zika?
We're currently working.
You're currently working. Laura?
Yes.
Charlie?
We're adding it to our flavivirus collection, yes.
All right. For you, Charlie, this must remind you of Hep C, which I remember you as a yellow fever person ages ago, more or less when I got started in virology. And then hepatitis C virus appeared, another flavivirus. You jumped on it and made that the focus of your lab, right?
Yeah, we certainly got distracted for a while from sort of the mosquito-borne flaviviruses to work on hepatitis C, which is really a much different beast. But so, I mean, I think thinking about Zika and sort of watching this field develop has been kind of a deja vu all over again, in the sense we have a new sort of member of the family that is getting a lot of attention, and fascinating biology, and a big problem. So.
Of course, you can grow it as opposed to hepatitis C. Right?
Yeah, no. Some people can grow it better than others, yes.
Yeah, it took many years to figure out how to grow Hep C in culture, right?
Yeah, 15 years.
All right, Laura, let's talk a little bit about mosquitoes. All right, but what are the ones we need to worry about in terms of transmission of Zika virus?
Well, the ones that, or the species that everybody's just sort of accepted is Aedes aegypti, partly because of historical records in Malaysia, and where there was an isolate, a single isolate from aegypti. There haven't been very many isolates, but the association of the mosquito with urban outbreaks points to Aedes aegypti. So of course that's a concern, and they're very difficult to control. So that's a major research effort, is novel ways to control this species. But the other one that people are worried about, particularly for the US, is Aedes albopictus. And they are competent, they can transmit, they get infected, they can transmit, but they haven't been involved except in Gabon. And that they were shown to be competent in Gabon, but they also only had a very low infection rate of less than, an MIR of less than 1. So we don't really know, because we don't know how the genetics of the African albopictus compare to the genetics of the US and the South American. Well, we do know some in the US. But the genetics of these mosquitoes vary tremendously. Also Aedes aegypti, I mean, it's most variable in Africa where it originated. And so you have to look at the genetics of the mosquito with the virus and do those kinds of studies. You can't extrapolate from one population to another. So those are the two main mosquitoes, and now there's also evidence of antibody in non-human primates in Brazil. So we have to think about sylvatic canopy-dwelling mosquitoes and how is this going to impact the persistence of the virus in the environment. So I think there's searching, people are looking, we know again the African Aedes that can transmit. There's a number of species starting with Aedes africanus where the virus was isolated, Aedes vittatus, which are competent vectors also, but we really don't know. The virus has changed a little bit. We really don't know what's going to happen in the Americas. Will the virus be sustained in the sylvatic cycle? How will that interface with the urban cycle? And Aedes aegypti, will they be the main vector as they are with dengue, or will we see some albopictus outbreaks in the U.S. where there's more widespread population of the mosquito?
So when you talk about vector competence and transmissibility, these are properties you determine in the laboratory, right?
Yes, vector competence is determined in the library, but in the laboratory, not in the library. But lately, it's been the library. But that's just one aspect of vectorial capacity, and that's what I'm going to be talking about tomorrow. I mean, it's a minor aspect, really. So we know with yellow fever, Aedes aegypti is not a very competent vector for yellow fever, but we have yellow fever outbreaks that are aegypti-driven, of course. We all know that. So it's vector competence, feeding, how often the mosquito feeds, are they focused on feeding on man? So Aedes aegypti, for example, one of the reasons they're such a threatening vector is that they feed almost every day on humans. They don't take sugar the way that most mosquitoes do. Most mosquitoes take a blood meal, digest it, and while they're digesting it, they're taking sugar meals, they lay their eggs. But aegypti will feed almost every day. So that one infected mosquito in a household can transmit to the entire household, theoretically. And that's the problem with control, is that even if you get the population down 90%, you probably won't control virus transmission very successfully. It really has to be almost elimination of the mosquito. So we do study vector competence in the lab, and many labs are doing that. But again, I think it's really important that people use uncolonized mosquitoes. We know with Aegypti from work that was done with dengue that after a few generations, they can't distinguish, for example, South American dengue, the American genotype, from the Southeast Asian genotype. And so Zika doesn't have these same kinds of genotypes yet, although the virus has changed. So we have to be careful about looking at studies that have been done and feeling, okay, it's done now, because people are mixing African virus with Asian mosquitoes, highly colonized mosquitoes with field mosquitoes. And I think we're more sophisticated than that at this point. We understand that it's the details that really matter. And then we have to look at the interaction of the mosquito genetics with the virus genetics, and then with the environmental factors. And it gets very complex in drawing a good picture, but you can do it to a certain extent in the lab. The one thing we can't get at in the lab, I think very confidently, is feeding rate, because we can offer blood every day, but it's not the same, and determine, okay, the mosquito's feeding, Aedes albopictus will only feed once every 5 days, but Aedes aegypti is feeding every day. But it's an artificial system, so we can't get at that. Similarly, survivorship is a really key component of vectorial capacity, because the mosquito has to take its first blood meal and get infected, allow that virus to amplify in the mosquito, get to the salivary gland so it can be transmitted in a second blood meal or a third blood meal. And that takes the length of time, the extrinsic incubation period is called. For the virus to do that, the mosquito has to be able to live that long. So that's very key, and that's also difficult to do in the lab, although we've done a number of experiments at different temperatures looking at that. And then the immature stages haven't been looked at very much. So these mosquitoes all breed in water. They breed in Aedes aegypti and albopictus breed in containers or tree holes for albopictus. And Aegypti are actually changing their behavior. Some of them are going underground. They've seen in Trinidad when the temperature goes above 34.5 centigrade, they'll start breeding in septic tanks underground, so that's going to be impossible to control. But we have to look at nutrition in the immature stages, competition, predation, and that's the other thing that a lot of labs are trying to get at, and that impacts the fitness of the adult. So it's not just that adult mosquito out there as a mosquito. There's so many differences, which is why we want to use field populations.
So West Nile virus came to the U.S. in '99, and you've spent many years studying that, and
Still are.
Still are. And in fact, in New York State, New York State established a screening program, correct, for West Nile
You mean
mosquitoes?
A screening program?
Well, it's called a surveillance program.
Oh, a surveillance program.
Sorry.
Yes.
So, are you? What is that? What do you
So, so New York State wants to know mosquito population levels, which are an important component of vectorial capacity, not the most important component, but it's important. Of course, if you have a low competent mosquito, but there's lots of them out there, it balances out. So they want to know populations of, in case of West Nile, and New York, it's mostly Culex pipiens. And it varies with where you are in the United States, which Culex species is most important. So, and they want to know the infection rate. So originally, states were also looking at bird infection rates. West Nile is different from Zika in that it requires the zoonotic cycle, the amplification in the bird-mosquito cycle. Birds amplify West Nile to very high titers, so that almost every mosquito that feeds on them gets infected, but they're not all competent. They don't all transmit. So we test all mosquitoes, but really Culex pipiens are the most important mosquito. And we also showed that they're important as a bridge vector, because what West Nile does, which again is different from Zika and Dengue and Chikungunya, is it has this zoonotic cycle, and then when it reaches a certain intensity of transmission, it spills over into other vectors and infects humans and horses, who don't have a high enough viremia to reinfect that zoonotic cycle, so they're dead-end hosts. Whereas with Chikungunya, Dengue, and Zika, they came out of a sylvatic cycle, but they don't need that sylvatic cycle to perpetuate. They can perpetuate human mosquito. So but labs, with some exceptions, birds are not regularly tested because we know that if there's West Nile there, they're positive, but it doesn't tell us what the risk is of infection. And West Nile is still a very important virus. I mean, people aren't thinking about it very much because they're so worried about Zika, but West Nile will cause more deaths, and I would predict, and illness in the US than Zika will.
I was just at the Bronx Zoo last week for a podcast, and they showed me Ground Zero, where the flamingos were first dying of West Nile.
Maybe Ground Zero,
Well,
maybe not.
It was very interesting because they have serum samples stored for years, and the veterinary director of the zoo told us that they were blaming them for the emergence of the virus. So they went back into their banks and pulled out all this urine and showed that they were negative.
Right.
Really wonderful story. So the question everyone wants to know is whether, like West Nile, Zika will spread throughout the U.S. And it seems to me that not having an animal reservoir mitigates against that to some extent.
Well, it's not having the animal reservoir, but it's not having large populations of Aedes aegypti except in border states like southern Texas and parts of Florida. And Arizona also has some locations with a lot of Aedes aegypti. And Aedes albopictus don't focus their feeding to the same extent as aegypti does on humans, although when Aedes aegypti are not around, albopictus will feed more often on humans, what I understand. So we don't really know, except from historically, we don't see dengue sweeping outbreaks in the US. We don't see chikungunya outbreaks in the US. There was some There have been cases of both in Florida and in Texas, but that are locally transmitted. But I would expect Zika to follow the same path. And if anything, the Aedes aegypti seem less competent for Zika to me than they do for dengue and chick, but I can't say that with any firmness. But the transmission efficiency is very low when you look at U.S. mosquitoes so far, what we've seen, except with the African strains. The African Zika strains seem to do quite well, but that's not what we have.
Mm-hmm. So you're concerned about extrapolating from the lab to what's actually happening out there. We don't know what mosquito is going to vector Zika in the U.S., right?
Well, we can say that it's going to be aegypti and perhaps albopictus, similar to dengue. We do know. Whether there are others, we don't know. There's been talk of Culex quinquefasciatus, which is the southern house. Its nickname is the southern house mosquito, and it's very prevalent in Brazil, in these urban areas. I mean, the other factor is socioeconomic. So we don't, generally our houses have air conditioning. They're more separated, the lots are larger. We don't have the density of population living close together that you have in Brazil, for example, or in Mexico. So even where we have the mosquitoes, that's why we haven't seen dengue outbreaks and probably won't see Zika outbreaks. Now, if Culex quinquefasciatus is also involved, which I don't know, we're still trying to get some results on that, but we haven't gotten anything very positive. Then you have another vector. Whether that will actually be an important vector, I would think probably not, just from what we've seen, but I don't say that with full confidence at this point. But that's the concern, that there may be other Aedes. Right. But again, we haven't seen that with these other Aedes aegypti transmitted viruses.
So when will we know answers as to whether Zika will spread in the U.S., whether other mosquitoes might be involved? What's the time frame? The next few months, you think?
June, yes, we will know. Yes.
Where
are we? In what month?
We're in
yes, no, I know.
End of May, right?
Yes.
So by the end of the summer, we'll know.
Yes, we will certainly know by the end of the summer.
So it was interesting, you said if you knock down the population of Aedes, it still doesn't help because one mosquito can bite and transmit within a whole family. So efforts to use Wolbachia-infected Aedes, which will knock down the population, but not fully, that's probably not going to help.
I think it all helps. Yeah. It just doesn't, it's not going to stop transmission. Right. I think anything we can do, Wolbachia or the genetically engineered males that Oxitec is using, where the progeny of the mating don't survive. Everything helps. The vaccine will help. I mean, with dengue, they've seen that the vaccine plus knocking down the mosquitoes together is the most effective, but everything's in trial right now in Brazil.
All right. Any questions about mosquitoes? No? All right. Well, if you have one, you have a Twitter question?
Yes. Someone wants to know why the fusion peptide is so well conserved among the flaviviridae.
Flaviviridae. Well, that's
that's not a mosquito question.
No, it's not, but that's okay. We can answer it, right, Michael?
We can answer it. I think Charlie could also answer it as well. So that peptide is critical for a step that all flaviviruses need to achieve in order to actually infect cells. So what happens is the virus attaches to the surface of the cell, and then it goes in an intracellular compartment called an endosome. And then in that endosome, it needs to get out of the endosome to go into the cytoplasm, where the cytoplasm it can actually translate and replicate. But in order to get out of the endosome, it needs to actually promote membrane fusion. And that occurs under conditions of low pH. So the endosome, as you go further in the cell, the pH changes. And in that pH change, there are structural transitions in the envelope proteins which coat the virus. And And what happens is the virus goes from this particular structure, which is a sort of a ball-like structure or approximate of that, and then undergoes a structural transition such that the fusion peptides become exposed and they form a trimer, and this actually then, that hydrophobic region of it can actually bind to the membranes of the endosome and allow a pore to form to allow the genetic material to get out into the cytoplasm. And every flavivirus does the same trick, the same pH-dependent structural transition in the envelope protein that changes it, turns out, from a dimer to a trimer, and that allows it to get into the cytoplasm. So that fusion peptide is exquisitely conserved because it has to undergo a very, very concerted transition to allow the virus to get into the viral RNA to get into the cytoplasm where to replicate. So if you line up all the sequences of all the related flaviviruses, there is very little, if any, genetic variation in that region at all.
And in fact, an antibody against this is broadly neutralizing, correct?
Yes, antibodies against it are broadly neutralizing. There's a problem with that in that most of the viruses actually shield that region when it's outside the cell.
Right, right.
And so some of the viruses are a little bit more susceptible to it than others. It turns out dengue is, and it has to do with the fact that the virus breathes as well as the fact that the virus is partially mature and immature, so on some parts of it, it may be exposed. Whereas other viruses like Zika and like West Nile actually are more like a ball, and the fusion peptide is buried and not exposed. And so in those cases, the broadly neutralizing antibodies don't work nearly as well as they do for some of the other flaviviruses.
Well, since we have your attention, Michael, you very quickly this year published 2 papers on a mouse model for Zika infection and then for virus crossing the placenta and replicating in the fetus. That's a mouse model. And my first question is, how did you do this so quickly? It's almost as if you were thinking about it last year.
He was.
Well, there are really 2 reasons why we were able to do it quickly. One is we were thinking about it last year, and in fact, I went to a meeting in last June at the NIH sponsored, which was a Chikungunya meeting. And as it happens, there was a number of investigators from all over the world that were there, many from Latin America, and a subset of those from Brazil. And while we were all talking about Chikungunya, which was, if you remember, a year ago or so, a very big concern propagating through Latin America. But while we were talking about vaccines and about the virology and about the epidemiology and potential therapeutic trials, at the breaks, we just sort of introduced ourselves and started talking, and there was a discussion going on amongst some of the Brazilian investigators that they were seeing some strange things with another virus at the time, which was Zika virus, which had been identified. And I talked to them, and they said yes, and we've seen these what seem to be birth defects, and the virus is behaving somewhat funny. We're not certain, is it this virus or is it another virus, but we have seen evidence recovering the virus at the time. And me being trained as a flavivirologist, although my lab does a lot of work on other viruses, still we have a very strong interest in understanding flaviviruses from a very basic perspective. Thought to myself, this seems sort of unusual and interesting. And the backdrop of that is that I started my lab, and Laura knows this quite well, I did a postdoc in dengue virus, but I started my own independent lab right at the time West Nile virus was occurring in the US. And immediately I started to work on West Nile virus as it emerged because there wasn't as much done and it was quite interesting.
Yeah, you shut the lights off.
And then I thought we were getting to, I thought Vincent was giving me a sign.
30 seconds.
So I remembered this, certainly, that as viruses emerge, and if there are viruses that you were working on, you're able to make a significant amount of traction. You have all of the tools, in some ways, how to grow the viruses, what kinds of animals might they infect, what will be key questions in the life cycle that you might want to answer. And so that's the first thing, how we got interested. So we started actually a year ago, last June, in doing this, and I had a very talented postdoc at the time, Helen Lazier, who's been on TWIV, as you are aware, in the past. This is prior to her getting her independent job at UNC, and she said that she wanted to start to work on this, and so we started to work on it together in our laboratories, and she has developed her independent program as well. And since that time, of course, as the number of cases started to escalate, as the global public health community became more concerned and more aware of what was going on, we started to escalate our efforts. And then I had a number of, key people in the laboratory basically say, I'm working on this project, but if you need me to work on Zika, I'll work on Zika too. And so there was a significant effort on people in my lab willing to sort of take time away in addition to their projects to really do this. And I had another talented postdoc, Jonathan Miner, in the lab who just said, I'll do it. If I'm here all night, I'll do it. And a number of people felt that way, and they've been working pretty much around the clock over the past 6 months or so to try to establish these models. So we made a lot of traction because I heard about it a year ago and decided as a flavivirologist I ought to be working on it. And then people in the lab really were inspired to actually put in the tremendous amount of effort to do it.
We had a press conference before this TWIV, and a reporter asked the panel, of which you were a member, why did we get caught by surprise by Zika? And we didn't, in fact. We have a trove of flavivirologists who are ready to jump in. And for the same reason, we were able to develop AIDS antiretrovirals very quickly because we've been working on retroviruses for so many years. So this is a great example of how virology needs to have a broad base of investigating many viruses, and then you can respond very quickly.
And I would just say that, anecdotally, many of us in the field don't just have one virus of the subtype. I know Laura has a freezer like I have a freezer with many flaviviruses, many alphaviruses. And so and have them and have grown them and understand, their conditions so that if something happened, we're ready to take it out of the freezer because we don't exactly know what the next thing will be.
Yeah. That resonates with you, right, Charlie?
Yeah, absolutely.
We have to have resources, and also we have to share them, of course, right? And be ready to work on something else. Now, let's talk a bit about your mouse models. One of the things you need to do is you have to use an immunologically compromised animal. Otherwise, the virus doesn't get into the fetus. Is that correct?
So it depends on the model. We've been establishing a couple of different models. If you are looking for the in utero transmission model, in our hands, we've had to immunocompromise the mother to some degree. There are other models. For example, if you infect the newborn animal, they do not need to be immunocompromised. They are sufficiently immunocompromised at birth that you can just infect them and they actually develop disease, and there's nothing that needs to be done at all. But in the in utero transmission, because the mother is immunologically intact enough, and we can come to why I think that is, we do need to suppress her innate immunity enough to allow the virus to replicate, to cause a sufficient level of virus to go into the blood to then get to the placenta.
So in these models, in which the innate immune system is compromised, right? The virus, when you inject it, I think you put it in the foot pad, correct?
Correct.
Does it replicate locally and then establish a viremia?
Yeah, so it replicates locally and then goes to the draining lymph node, and then it seeds the bloodstream and then can go to different organs, the spleen perhaps. Sometimes some of the viruses will also go to some of the other organs. For example, we've seen virus go to the testes, to the eye, as other sites. And then, not in the mother, of course. In the mother, there's no testes, but in the mother, the virus will go to the placenta and then replicate there. Now, in our in utero model, the fetus was actually not immunocompromised because we crossed the mother to a wild-type animal. So the fetus was heterozygous. So then the virus managed to cross, after infecting the local cells, which are called trophoblasts, could cross the trophoblast barrier and then get onto the fetal site, actually infected the fetal endothelial cells, and then would spread to the fetal brain and cause injury there, in addition to the injury that was happening in the placenta.
So do you think the kinds of changes you see in the infected fetus are mimicking what's going on in infected humans?
So that's a challenging question. We are still learning. This is a model. I think there are, if you look at it, certainly there are some of the things that are similar. I'm not certain everything is going to be identical because it's a model for it. We certainly see evidence of fetal infection in the brain. We see evidence of cell death, massive cell death in the brains as well. That is, we know those are occurring. In the in utero model that we didn't see microcephaly per se. What we saw more of, which was placental insufficiency and what's called intrauterine growth retardation. Now that is seen in humans. And you can see in the clinical trials that have been done in Brazil, they have seen a number of women who've gotten what's called IUGR, intrauterine growth retardation, and that's led to fetal demise or spontaneous miscarriage. However, of course, the press has picked up on the microcephaly, which is prominent enough. In our mouse models, we didn't see that. But we think that maybe because of the timing of when we actually injected the virus, which was very early in the first trimester, and we harvested the fetuses very early. And the mouse is a little different. There's a lot of mouse neurodevelopment that occurs after birth, more so than in humans. And it may be that if we infect later, we will actually see that. And in fact, another group had published a study concurrently with ours in a different journal, and they infected a week later, and they reported they were able to see both intrauterine growth retardation and microcephaly in that study.
I think there was another group that also injected directly into the fetal brain.
Yeah, that was an interesting study.
And there they saw microcephaly.
There they saw microcephaly and lots of damage.
Yeah.
I think Laura and I were talking about this a little earlier, and I think there's certainly validity in understanding what could happen if the virus gets into the brain. But certainly it is artificial to introduce the virus directly into the brain in utero. And it's sort of like what we do sometimes in adult animals. We might put the virus through an intracranial route to ask the question, is the virus that we're studying, maybe it's a genetic variant or a wild type virus, is it neurovirulent? Can it kill neurons? And that's an important test to do. In this case, it was used in this fetal model, but it was clearly an artificial model because it bypassed the mother entirely.
Mm-hmm. Yeah, and you want to know those early steps, what's happening,
Correct,
of course.
because those are all steps possibly for intervention.
Now, why do you have to immunologically compromise the mice? The mothers are presumably not immunologically compromised to the same extent. I mean, the mice are lacking interferon receptors in your case, right?
Either interferon receptors or we give an antibody to
right.
partially block interferon. The reason is probably what we've learned from dengue virus, actually, from studying another flavivirus extensively, not just my laboratory, but many other laboratories in the field, in that for viruses to cause disease in an animal, or even to replicate to high titer in a cell, they need to overcome the cell-intrinsic immunity. And that much of that immunity, although not all of it, is due to interferon. And so what viruses do is they figure a way with their limited genetic capital, or if they're inefficient pox viruses with lots of their genetic capital, they figure a way to either evade or actively antagonize that part of the immune system. But they do it in a species-specific way often. So for example, Zika virus, like dengue virus, really occurs in humans and perhaps rhesus macaques or some other monkey models. So it's not really a mouse virus. So it really doesn't care about the mouse immune system. So what people have already found, and this is work that's been now published by Adolfo Garcia Sastres group just last week in Cell Host and Microbe, they showed that the viral NS5 protein, which is one of the non-structural proteins, can actually promote degradation of STAT2, which is a key signaling molecule downstream of interferon. But it does that for human STAT2 and not for mouse STAT2. And dengue was the same way. It antagonized human STAT2 but not mouse STAT2. So in other words, there's a species barrier, and this species barrier, which actually was seen for many other viruses, in fact, Grant McFadden, who's in the audience, saw this for pox viruses years ago, determines whether a virus may be able to actually replicate efficiently enough to cause disease in an animal. And if it doesn't have that species-specific evasion mechanism, it's going to be restricted. But so we took away the evasion mechanism, and then this allowed it to replicate the higher
So you would
levels.
predict that in monkeys, the viral protein could antagonize monkey STAT2, right?
You would predict that. I don't recall Adolfo's paper that he actually directly tested that, but there is now data, as you're probably aware, people are streaming online, the University of Wisconsin, data in non-human primates. And there's data that's suggesting that you can get persistent viremia in some of those monkeys, in pregnant monkeys, for example, as well as getting high levels of virus replication in the non-pregnant monkeys. And so I would predict that it would be able to antagonize monkey STAT2, yes.
So what do you hope to learn from these mouse models?
So there are a lot of questions that we're interested in, whether they're the in utero transmission model or not, or the neonatal, the newborn model, or even the adult models as we're looking at them. Questions that we're interested in are how long does persistence go on? Mm-hmm. What is the long-term consequence of persistence? How is persistence actually happening at the molecular level? What aspects of adaptive immunity, especially B-cell immunity? In my laboratory, we've had long-standing interest in understanding how antibodies neutralize viruses. And although we would think that it should be the same way as other flaviviruses, like dengue or like yellow fever or like West Nile virus, really needs to be actually addressed whether it is or not. We know the viral particle is a little different. So there was a cryo-EM paper that was published, as you're aware, I think you may even have covered these on TWIV a few weeks ago, showing that the virus is a ball, and it might be that there's some greater contacts between some of the envelope proteins. How that might impact antibody neutralization, which is important for either antibody-based therapeutics or vaccines which target particular epitopes. Those are the questions that we want to ask. And in the in utero model, we're still really trying to understand the cell types, what determines it, what is the intrinsic immune responses that can actually limit this, and are those particular points at which you could actually then target either small molecules or for therapeutics, or even just learn about the basic biology and how it is different than perhaps the other flaviviruses, which could explain its tropism.
So you probably have also thought about the problem of antibody enhancement, either antibodies to dengue. So we know that dengue antibodies will cross-react with Zika. What other flaviviruses do we have to worry about? Tick-borne encephalitis, is that another one that has cross-reactive antibodies?
So it depends, and it depends on the species. There's been not as much data generated with the other flaviviruses, for example, in humans, to know what the repertoire is. There is certainly good data now for West Nile from a few groups that have been published. Dengue, a lot of data has been published about this. And then there is very little data on JEV in terms of what is at the monoclonal level, what are the repertoire analysis. And there's a little bit of data from TBEV from Franz Heinz's group in Vienna. So depending upon the person and the virus, you may make more or less of these cross-reactive antibodies, which target the fusion peptide, which was alluded to a little bit earlier. And those seem to be the key ones which could actually be around in a person who is flavivirus immune or received the vaccine and is immune to that particular virus, which could, in theory, either augment Zika or maybe the Zika virus could augment them. Remember that the thing about immune enhancement and antibodies is it's been largely a one-directional process, meaning it's antibodies against dengue that have modulated dengue pathogenesis. Maybe there's some data that's out there that's very weak that suggests anti-JEV antibodies could do this in Asia, but it's never been shown definitively in humans that any of the other virus antibodies can modulate another virus, meaning, as an example, dengue antibodies modulating West Nile pathogenesis.
Have people looked, though?
People have looked in the lab for certain. Have people looked epidemiologically? I think less so. Some of the other viruses are not nearly as high prevalence, so they may be a little bit harder to do this. But Zika is closer to dengue, and so there is certainly a concern that either, as we talked about at the press conference, that Zika antibodies could modulate dengue pathogenesis, because it's sort of like a related virus, like another serotype of dengue, although it's more distant, or dengue antibodies could modulate Zika pathogenesis. That we can test in the lab, and we're actually doing that. There's still ongoing experiments, and so we don't know.
Seems to me this needs to be sorted out, because we do have a dengue vaccine which is being deployed at the moment. People are talking about a Zika vaccine, and they may actually cause.
But we needed a model.
Yes.
Right. So we needed to test. We needed. So we as you alluded to, the pace. Well, maybe we haven't alluded to that. The pace of Zika virus research over the past several months has been remarkable, absolutely remarkable, and not just from the U.S. Globally, China, Latin America, parts of Europe are working on this now. And so every day or every week there's just new papers coming out. And so we had to set a model up for an in utero transmission model to see whether now if you either passively transfer the antibodies or if you vaccinate the mice, the mother mice, and then see what happens. And so those studies just are ongoing now. But without an animal model, it's actually difficult to do this.
I guess we'll see them next week in Cell, right?
We'll see.
Michaela, your specialty is innate immune sensing of flaviviruses and antagonism of it, correct? And so a lot of what Michael has been saying about antagonism of STAT2, you're very familiar, but you've discovered some other means of antagonism, right? Tell us about those.
So we recently identified that dengue and also West Nile virus uses the NS3, non-structural protein 3, which is actually the protease of these viruses. So it's an essential enzyme which is actually responsible to cleave the polyprotein into the individual proteins. But this is the same protein used to antagonize one of the major sensors in all of our cells in the human body, which is called RIG-I. So this is a viral RNA sensor. So if cells get infected with all kinds of viruses, including dengue and West Nile and also Zika, then RIG-I recognizes the viral RNA, and this is really the first molecule which then in the end recognizes there is a viral or a virus now inside the cell, and then initiates an innate immune response, leading to cytokine production, especially type 1 interferon. And most viruses, I mean, we have worked previously on influenza virus, also on paramyxoviruses. They have really sophisticated ways to block these innate immune sensors, including RIG-I. And even though we know actually for several years now that RIG-I is a major sensor for dengue and also West Nile, in general flaviviruses, it has been largely unknown how dengue and West Nile virus evade the RIG-I dependent response. We know certainly other innate immune pathways which are suppressed.
So the one
by
you were
dengue.
talking about is downstream of
Correct.
interferon
Correct.
production itself, the induction of the interferon stimulated genes, right?
Yeah, in fact, flaviviruses use multiple proteins at multiple
Right.
steps to disable the key innate
It's
defenses.
amazing we're still
And
here.
Michaela has been identifying, one of the key pathways, but they target many steps, not just one. They don't leave
Okay.
it to chance.
Actually, it's most viral pathogens,
Yeah.
and also the flaviviruses.
Well, I
they
always say
target.
that if you don't target, if you don't antagonize, you're gone, right? You're eliminated.
You're not
Correct.
nothing we're talking about.
Right.
Yeah. Okay, sorry, go ahead.
So we
RIG-I.
found that the protease actually inhibits the sensor RIG-I in an enzymatic independent manner, which is different, for example, to hepatitis C virus, which uses the very same protein, however, cleaves a critical signaling molecule in this pathway. But dengue and West Nile virus, they use the protease to block the translocation of this sensor from the cytosol to mitochondria. Maybe I should explain, the viral RNA is usually recognized in the cytoplasm, but then RIG-I must translocate to an organelle called mitochondrium to then initiate signaling, which then leads to interferon gene expression. And really this step is basically blocked by the NS3 protease and it's not cleavage dependent or anything, but we identified a 4 amino acid phosphomimetic motif, which is conserved among dengue, West Nile virus, and we actually also see it in the Zika NS3 protein, which is responsible for actually targeting a trafficking protein, I call it, called 1433 epsilon, which brings RIG-I from the cytoplasm to the mitochondria to, in the end, initiate signaling.
We have all these beautiful pictures of signaling arrows, and we never know how the arrows work, but now we know at least
like that.
something is bringing it. Things don't just diffuse. So this protease, you're talking about dengue, right? Does it also, from Zika, does it also antagonize the same cellular protein? What's the name of this? 1433?
1433 epsilon. So actually, Michael Gale's group here identified that this is a protein which brings RIG-I from the cytoplasm to mitochondria. So we see in Zika the very same 4 amino acid phosphomimetic motif, and we have some preliminary data that Zika also can bind efficiently 1433 epsilon.
And we'll see that next week in Cell 2, I presume. Michael, didn't you check a RIG-I knockout mouse with Zika, or did you not?
We checked a MAVS knockout mouse.
MAVS.
The RIG-I knockout is problematic because when you breed it to purebred mice, it dies. But the MAVS knockout,
It is problematic.
yes, it's problematic. But the MAVS knockout actually, those animals live because MAVS is only one of the pathways by which you can generate interferon. It's a dominant pathway, but there are bypass pathways through other sensors. And so the mice did actually get sick and did show some evidence of weight loss, but they didn't actually die.
Okay. So you're looking at this currently for Zika to see if epsilon 1433 is antagonized, right?
Yes, and we also, in our publication, we generated a mutant dengue virus in which we eliminated this host virus interaction. And usually what in the end came out and what we expected is that you have a strongly growth attenuated virus, because now this virus cannot evade a major innate sensing pathway, meaning this mutant virus is now readily recognized by our innate immune system. So you see efficient cytokine production and antiviral gene production, and thereby it has a relatively slow or reduced growth compared to the parental virus.
Of course, unless you grow it in a immunocompromised cell or animal, then
correct.
it should be fine, right?
That's basically how we produce usually these mutant viruses. So we grow them up in viro cells, which are known to be interferon effective, or also mosquito cells, which have certainly are defective in the interferon response. That's basically how you generate such mutant viruses in efficient titers.
So if you were able to generate a Zika virus mutant of this sort, would that be a potential vaccine candidate?
Well, I'm always, I would be careful with that. I mean, in general, there's always, if you generate mutant viruses, there's the risk of reversion. So that happens, can happen in vitro, certainly in vivo in mouse models or even in humans. So certainly you would want to prevent that. Otherwise it reverts back to the wild type and then you have a fully virulent virus. So I don't expect that our mutant virus, in which we mutated actually made two amino acid changes. It's actually on nucleotide level 4 mutations, but certainly it's too much of a risk that it could revert. But our work is more like it provides a principle. First of all, it shows us really the biology and immune evasion of these viruses. But it could be in the future, I think, if we eliminate not only the 1433 NS3 interaction, but maybe we include also STAT2 and NS5 interaction and maybe 5 other immune evasion mechanisms. As Michael said, there are multiple different proteins involved and also host proteins targeted. So maybe if we could in the future rationally design some attenuated viruses by eliminating several of these immune evasion mechanisms, that could eventually, you see already how I'm talking, eventually lead to.
But you don't want to make it so attenuated that it doesn't boost your immunity.
It's always a balance.
So this whole problem with reversion is very interesting because years ago Albert Sabin made a polio vaccine. He had no idea what mutations were in it, and it reverts because there are only 2 or 3 point mutations that distinguish it from the wild type virus. But now we can sequence these genomes and know exactly what's going on. So do you think of the innate response as a target also for antiviral therapy at all? I think some people do.
Yes, this is a completely different concept. So usually we try to target the viral protein. Of course, you put basically selective pressure on the virus, meaning then usually the virus can mutate. So this is what we have in many different, like for HIV, for example, where then the virus escapes. So another strategy is really maybe to find ways to boost our innate immune system, to make it more efficient. And people are working on that by certain agonists for RIG-I or other ways. And we have done work on certain regulatory proteins, which are important for an efficient innate immune response. So their activity could be enhanced or modulated by small molecules or other means to find ways to boost our innate immune system.
So what remains the focus of your lab? Do you think it will, just like Michael transformed into a West Nile lab, will you transform into a Zika lab, or your focus is dengue at the moment, right?
Yeah. Well, I have to say, actually, the research in my lab is really focused on innate immunity.
Okay.
And we have done work actually, I have to say I'm a newcomer to the flavivirus field, so we have done work on influenza A viruses. We are currently working still on influenza virus, also on herpes viruses. I have to say that's my background, gamma-2 herpes viruses. But we are currently working because we are really fascinated by this conserved, immune evasion mechanism with the NS3 protein and 1433 epsilon. So we are really a major focus of my lab are now these 3 flaviviruses, trying to understand how they evade innate immunity.
Thinking about this evasion and species adaptation that you mentioned, one could take all the target proteins, either 1433 or whatever else the target is, among different species, and simply ask, can they be evaded by a particular viral protein? And that would give you an idea of the host range of viruses, right? I'm sure people are doing that. There are people like Harmeet Malik love to do that sort of thing.
So you can do it at the computational level, but the sequence gazing level at what I'll call 30,000 feet.
30,000.
Or you could be the old-fashioned biochemist and ectopically express it in cells and do pull-downs and see what pulls down and what's missing. So you have your choice.
Yep. We're in good times.
Question, does the NS3 Michaela, does the NS3 mechanism also work in mice?
So
or is it species restricted?
It's different from, for example, the NS5 STAT2 mechanism. So 1433 epsilon is identical on amino acid level to the human, between the human and the mouse. So it's identical, the protein. It's
and
really
you're sure it's a direct interaction?
Yes. So we have data. Also, actually, we are collaborating with Mike and have generated already a mutant West Nile virus, and it seems to have actually great relevance in vivo in mouse models, so it's targeted also in mouse based on our data. Yeah.
And have people made a sort of a human STAT2 knock-in? And does that
I heard about it last week.
does that now sort of overcome the need for?
Vincent.
I was on a coffee line and someone said, yes! And I said, and that was the result, yes. But I can't tell you who it is. I shouldn't because it was on a coffee line, you know. But yes, it's been done and you would predict the result. You knock in the human and it makes the mice.
Unless there are multiple ones. So for dengue, for example, there's STAT2, but there's also sting, and that sting also is species specific.
Right.
So there may be actually multiple innate immunes, some of which are not species specific, and some are, and you may need to make them additive. We just don't know.
In this case, the knock-in of the human STAT2 made the mouse susceptible to infection. So you don't have to have an interferon mutant mouse. So that should be out next week in Cell.
Okay.
This is a theme, right? Charlie, you spent many years trying to figure out how Hep C enters cells. And it requires many different proteins. Too many, you know, I work on polio where it needs one protein to get in.
It's probably using other factors, right?
Uh,
A principal receptor, yes, but
I don't know, you can put poliovirus receptor into many, many different cells of different species and the virus gets in unless they're all conserved. Anyway, it looks like Zika might require multiple entry factors as well. There have been a number of candidates. I wonder if you thought about that at all, or perhaps you're working on it.
I mean, we haven't.
You had enough of multiple entry
Yeah,
factors.
we've had enough with multiple entry factors. No, I think that, I mean, that's one of the great sort of challenges, I think, in the flavivirus field is, you know, sort of what is the sort of entry mechanism that they use? Is it a common mechanism among, you know, sort of different groups of flaviviruses, and is it a common mechanism between sort of insect cells and vertebrate cells? So I think people have some sort of thoughts on what might be molecules that are involved in Zika, but I think at least so far, at least the data that I'm aware of are pretty preliminary. So I think there's still quite a bit of work to do there.
Okay, would you agree with that it's pretty early days in terms of receptors?
Yeah, I'll just say that, and I think it's true for all flaviviruses, not just Zika. And you know, my original postdoctoral project, I don't know whether Laura remembers this because we were both in California at the time, was to identify the dengue receptor. I failed in that one. And there's been some attachment factors identified, but a bona fide receptor is the way you think about a poliovirus receptor. Or HIV has not been identified.
Right.
I will say one other thing is that our lab, along with a couple of other labs, have done CRISPR screens, whole genome CRISPR screens, to identify host factors required for flavivirus infection, including Zika virus. And we did not identify an entry factor for Zika virus in that process. We identified some other things, but nothing on the surface which would stand out to say, just like Charlie has done elegantly with, you know, Claudin or Occludins or CD81 or anything like that. Nothing like that at all.
Were you in the same lab as he was in California?
No.
He was a faculty member.
You were faculty. Okay. All right. Charlie,
But we
you-
did do some work
Sorry.
together.
That's right.
Where was this?
At Berkeley.
Berkeley? Okay. I forgot to find out where you guys were trained and all that, which I usually do, but maybe we'll do it at the end. Charlie, you've touched on so many aspects of fundamental virus replication with the flaviviruses, so I just want to go through some of those. We touched on the receptor. One of the culminations of work on Hep C was development of antivirals based on fundamental knowledge of the replication cycle. How is that going to go for Zika, do you think?
Well, I think, you know, there has been a lot of effort over the last decade to look for flavivirus antivirals. And I think in the case of hepatitis C, I mean, it's actually kind of ironic in a way, because I think one of the aspects that we began studying in hepatitis C was the viral protease, the serine protease that Michaela talked about, which is, you know, shared with hepatitis C and then emerged as, you know, a very good target for antivirals after a lot of effort. I think Pei Yang is going to talk about this tomorrow in our day on Zika, but you know, right now there are no, you know, sort of approved small molecules for treating flavivirus infections, and I think that they will come. I think there are already some some hopeful compounds out there. And I really think we do need to make a push to sort of develop some that are safe and effective just to see how they would enter into clinical use. And because I think it's somewhat difficult in some scenarios. I mean, hepatitis C is a chronic infection. I mean, you know your target. You know, in Zika or dengue, you know, sort of when people come in, you know, the sort of viremic phase may be over, and you may be looking at, you know, sort of downstream consequences. So people could envision using these in a prophylactic way, or if infections are identified very early. I mean, the added challenge with Zika is that you want to be able to put this into people who are thinking about getting pregnant or who are already pregnant in a Zika endemic area. And so I think that is a very high safety bar. And so but I hope the efforts continue. I think got a lot of good targets. I think there are going to be a lot of interesting host targets, but also we already know the sort of key players that are important in the nuts and bolts of flavivirus replication. So I'm hopeful now more than ever before that people are really going to make a final push and come up with things that can go into the clinic and be tested.
Problem is, of course, that for most acute virus infections, you need a good diagnostic to be able to prescribe in time, right?
Right.
And from what I'm hearing, we have problems with Zika diagnostics, especially distinguishing it from other.
I think there's a lot of effort in that area to sort of come up with a more, you know, reliable IgM assay that would be more Zika specific. And of course, the nucleic acid-based assays are certainly specific. Maybe they don't have the sensitivity quite yet that one would like for blood screening purposes or whatever. But I do think that, I mean, the problem with the nucleic acid-based diagnostics is that this virus doesn't seem to be like yellow fever that, you know, in a vertebrate host can reach, you know, 10 to the 9th in circulation. Zika seems to have somewhat lower titers, at least based on some of the data that are out there, and it seems to be very transient. So you have a relatively short window in acute infection where you can even detect viremia. So people are looking in saliva and semen and urine.
It lasts much longer at higher titers.
And also part of the problem is that there's a high percentage of asymptomatic infections, right?
80%.
80%. So do we know actually if you are asymptomatically infected, do those, if you're pregnant, do you get birth defects? Do we have any? We don't know at all.
No.
I thought there were some cases that have been noted.
We don't have any numbers on frequency.
No numbers, but there have
The
been asymptomatic.
Infections that resulted in
we have
birth
no
defects.
idea
Right. What is the overall, I know it's early days, but what's the overall percentage of infected pregnant women who will develop birth defects? Do we have that? I think you guys said 1% earlier at the press conference.
Yeah, that it sort of depends. In the study that was published in the New England Journal of Medicine a few months ago by Brazil et al., they showed that 29% of the women who had a rash, who were then diagnosed as being Zika positive, 29% of those had some congenital abnormalities on ultrasound, ranging from, you know, relatively small ones all the way through microcephaly and newborn to fetal demise and miscarriage. That's a very high number, and certainly doesn't address the asymptomatic ones either. So we don't really know the denominator of that, but the number that's been thrown around is about 1%.
And that comes from French Polynesia.
Correct. Retrospective analysis.
Retrospectively.
Was there a question?
So,
Stanley Malloy on Twitter says,
Stanley.
so maybe extensive mosquito transmission is not probable in the US, but what about the shift to extensive sexual transmission?
Wow. Who wants to tackle that one? Sexual transmission increased, because right now sexual transmission is low, if any, right? Personally, I'm not terribly convinced because there's virus in other fluids and that hasn't been ruled out. But let's say it's low. Could it increase?
I don't think we have enough data one way or the other, but clearly most of the cases, the vast majority, are felt to be mosquito transmitted at this point in time. What we have not done, for example, and we need a longitudinal study to look at there's two questions related to, let's say, what we only know so far is male to female transmission. I don't think there's any documented cases, not that it's easy to document a female to male.
That's correct. Yeah.
But only male to female. So
Male to male.
There's one
and male to
one
male as
to
well.
male,
Sorry,
yeah.
male to male, male to female, but not female to male. So I think two issues are: one, how long is there infectious virus in sperm? That's one question. A longitudinal study of many men to follow this out and see for infectious virus as well as RNA. And then a second one is, what is the threshold amount that is actually necessary to transmit? And I think based on that data, as well as good epidemiologic data, where there are no mosquitoes around, we could begin to address the question. But it is very difficult to speculate, other than most of the cases right now can be accounted for by non-sexual transmission.
Stanley's being provocative, yeah.
I think
Okay.
the longest period of time that's been seen is 62 days in the semen. Yeah.
Which is a long
Is there
time.
another question?
Yes, Aaron Hodge on YouTube says, superbacteria versus Zika virus, who would win? I thought you'd like that.
Different sport.
Apples and oranges.
Apples and oranges. Charlie, one of the things you did, which is very interesting, was to look for Hep C-related viruses in other non-human animals. So you found Hep C-related viruses in dogs, I think, horses. Is anyone-
Rats.
Rats?
Well, this is work from Ian Lipkin's
Yeah.
lab. Yeah, I mean, I think that, for many years we were sort of looking for an animal model, a la Mike Diamond could, sort of work on hepatitis C, and really spent a lot of time trying to sort of re-engineer mice to put in human factors and get rid of some of these innate immune pathways that were incompatible with this human virus, and managed to get a model that kind of limped along in terms of hep C infection. But I think with next generation sequencing, we're seeing our whole world of virology kind of being sort of expanded beyond exponentially. And in the case of hepatitis C and the passivviruses, there was one close relative whose origin was really not well characterized and has never been seen again, a so-called GB virus B. And then, yes, Ian's group found a more closely related isolate in a respiratory outbreak in dogs, which turned out to not be a common dog virus, but was actually found quite commonly in horses. About 30% of the horses are seropositive, and a significant fraction are viremic. And so it was renamed from canine hepacivirus to non-primate hepacivirus. And this, is a model that we began working on in horses. Still early days, and then the sort of latest. There was an intervening sort of wild rodent hepacivirus that was found in deer mice, and we were really excited about that. Got the virus, tried to infect laboratory mice, and even if we used ones that were interferon receptor knockouts and so on, it didn't go. But it turned out that the whole time, the virus that we really wanted was scampering around the subways and streets of New York. And Ian and his team sort of went out and trapped about 120 Norway rats, wild rats in New York City, and swabbed them and sliced them and diced them and sequenced them, and found 2 lineages of Norway rat hepacivirus. And this turns out to be, a fantastic virus for infection of laboratory mice. And you can establish chronic infections, and hopefully we'll be able to understand a little bit more about hepacivirus pathogenesis. But aside from that sort of microcosm of hepaciviruses, if you look at some of the data that people are finding when they start grinding up spiders and cockroaches and sort of looking at sort of what's out there, I mean, there are literally immeasurable numbers of viruses that sort of are closely or much more distantly related to our sort of favorite viruses that we've classified in different families and genera. And so I don't think, and we have no idea sort of what the species restrictions of these might be, and really when sort of the next one might show up and be a problem in humans. I think it bodes well for the longevity of
But in virology in general, I was interested because I wanted to know if there was anything related to Zika in any of these other species that have been looked at, but I'm not sure if we.
Well,
know
I
yet.
guess we've already got more flaviviruses than we can deal with,
Yeah.
with 70 or 80. But my guess is that as people do more next generation sequencing,
Yeah.
we'll be finding. And I think our picture is always sort of incomplete of what's out there in nature. And my guess is that there may be intermediate isolates sort of between, Asian or the two African sort of clades.
Right.
And
Or people have used the sort of deep sequencing approach to all these samples to sort of identify the parental ancestor viruses, like these segmented flaviviruses.
yes.
right.
Yeah.
Exactly.
So we have a segmented flavivirus
yeah,
from
which are
mosquitoes.
absolutely fascinating.
And there's one from ticks. From, I guess
Yeah.
Ian was part of that too.
So, monopartite, forget it.
Yeah.
And I've worked on one virus my entire career.
I don't know.
There are some of us like that.
Rumor has it
You have a
that you
question?
were,
Yes.
About the murine model, have you investigated any uterine DCs for interaction with the Zika virus and possible transmission?
So we have profiled a number of the immune cells in the uterus, and we've also looked to see whether they have been infected using either antigen staining or RNA fish staining to some degree. But we have not taken them out ex vivo, harvested them and infected them. There are other groups that are doing this. In fact, the person sitting in front of you, Mehul Sutar, sitting in the audience, has been interested very much in studying dendritic cell interactions with Zika viruses. So we've not been focused on that. Other groups are working on it, but we have profiled them to some degree.
Yes.
So this panel, and I think the series in general, have really suggested the strength of the academic community in advancing virology and responding to the urgencies that have appeared over the decades. I'm interested in knowing what you 4 or 5 think about the robustness of the commercial sector in being able to respond and bring products to market.
All right. Michael. If you don't know, you could say, I don't know.
I would say two things. One is, in times past, the commercial sector has been much more involved, I think, in basic research. The basic pipelines of products, they had their own research institutes. Some of them still do to some degree, something like a Genentech or some other companies, but a lot of that basic research that was done by companies, which was contributing to the fundamental knowledge, many of the companies have cut back and are not doing nearly as much of that. In terms of how the pipeline goes, I think that, as an academic, I have to say I sort of separate myself a little bit from the process, although we do interact with companies that are interested in vaccines or therapeutics, especially against viruses that we work on, including Zika virus. And so I think that there's a tremendous amount of enthusiasm and a response, an important positive response. In terms of what their impediments are, in terms of setting up clinical trials, the cost of actually getting a drug to market, dealing with the FDA, implementation, all of that stuff, the costs are very high. And maybe Charlie can discuss this a little bit. He may have some other knowledge, but I think that those are certainly issues that confront corporate America in dealing with trying to get products, especially for viruses, maybe not Zika only, but viruses that may be in the developing world where the product margin may not be obvious.
Thoughts?
Well, I think the scorecard is a bit mixed. I agree with Mike in the sense that we do see a lot of commercial entities that are de-emphasizing their sort of research pipelines and relying on academia to sort of do fundamental science. And at the same time, we are also, I think, as sort of basic scientists, being pushed by the NIH to do, more translational stuff. And I think these things are all important, but you have to have a balance. If you get too translational before you're ready to translate, you waste a lot of money. In the case of hepatitis C, where there are billions of dollars to be made, there was really no problem, at least after the first 5 or 6 years post the discovery of the virus, to get commercial entities interested. Although they were a bit slower to get interested in hepatitis C than I thought. It was the sort of diagnostics was the initial driver, and then the antivirals came later. But if you have a $11 billion annual market, then they don't have trouble sort of with the clinical development costs and so on. So I think it really depends, and I think for these arboviruses, again, it is an issue of what's going to be commercially successful, when you're talking about commercial entities. And I think there is always going to be this sort of gray area where it's really not in their financial interest to tackle some of these problems, but we really need to, as a society, make sure that we have the infrastructure and in academia and maybe in smaller biotech to sort of bring things forward.
I think in diagnostics, though, so when West Nile came to the US and it was recognized that there was blood transfusion transmission, the companies jumped right on getting a test, and FDA facilitated it so it could go through quickly, which is unusual. But so 2001, all these companies were working on a sensitive, specific assay to protect the blood bank, and it was implemented before the tremendous increase in transmission that occurred in 2002 and then in 2003, and had that not been in place and it was all done by the companies, it would have been very serious for the blood banks. And now with Zika, there's a lot of companies working on diagnostic assays, and we get involved as a state health lab because we have so many samples. They need to confirm and validate their assays. So I have to say some of them have been not very good, and they're out there on the market, which is a little frightening. But we've tested 5,000 samples in New York State to date, and so we have a lot of positives. So they do come to us, and those are all company-developed diagnostic assays.
All right, I know I'm the only thing between you and cocktails, so just have one more thing I want to do, and that is Michael and Michaela, Laura and Charlie, tell us just briefly about your training. Where are you from originally?
I grew up in New York City, New York.
Wow. And you went to college?
And I went to Columbia University, New York.
Wow.
Didn't know that.
When did you graduate?
I think, were you a faculty then? Let's see. I graduated in 1985.
Yeah, I was there for 2 years. Yeah. All right.
And then I went to Boston, to Harvard, and did a combined MD PhD degree. My PhD was in immunology.
Mm-hmm.
And then I went to Berkeley and did a postdoc in fly genetics. And then went and did a internship residency and clinical fellowship in infectious diseases. And then did a second postdoc in virology. And then started my own lab in 2001.
WashU.
WashU.
All right. Michaela, where are you from?
So I'm from Germany. So I grew up in Bavaria, about 2 hours away from Munich.
Mm-hmm.
And then, so that's where I did my undergraduate studies in virology. Then I did my PhD at Harvard University and did a quite short postdoc at USC, and then was hired back to Harvard, was then instructor there and assistant and associate professor, and just recently moved 8 months ago to the University of Chicago.
I did the first TWIV in Europe in Munich.
Okay.
They have a grant to do meetings, and they arranged the whole thing. It was great, and but we just missed Oktoberfest.
Too bad.
Laura, where are you from?
So I'm from New York, and I went to college at University of Pennsylvania in Philadelphia, and then went to Panama for a postdoc and studied yellow fever, and then briefly Madison, and then went to Berkeley to work on St. Louis and Western equine encephalitis, and was there until we moved the lab to UC Davis, and then in 2000 when West Nile came, I went to New York to set up the lab to study West Nile, and I've been there ever since. And when the Henrietta Lacks book came out about HeLa cells, I saw Chester Southam, who is somebody notorious, I guess, but I actually worked for him for 2 summers when I was in college. I had a position in his lab, and I thought, wow. And then he also inoculated cancer patients with West Nile virus. So I've sort of come full circle.
He was in the book because he inoculated people with HeLa cells. Right?
He was in the book because he inoculated people with HeLa cells to see if it was infectious and they would get cancer. But he also inoculated cancer patients with West Nile because they saw with Russian spring summer virus.
And did it work?
It didn't really work, and it worked a little bit with some, and a couple died. Things were very different then, but then the HeLa cell story was very interesting.
They're making a movie of that,
Oh, are they? I like the book.
Oprah Winfrey is going to star as Henrietta Lacks.
No.
Yes. Charlie, where are you from?
I'm from California. Did my undergraduate work at UC Davis, PhD and postdoc at Caltech, a brief sabbatical in Australia at Australia National University, and then WashU for 15 years, and then Rockefeller for 16 years.
Charlie and I did the first sequence of an RNA virus genome, not too far apart, right?
Yeah.
And we still talk to each other. All right, this is TWIV 392. You can find it at iTunes at microbe.tv/TWIV, on Google Play Music, or on your favorite podcatcher for iOS or Android. If you have questions or comments, please send them to twiv@microbe.tv, and consider supporting TWIV and our other science shows. Go to patreon.com/microbetv. I want to thank everyone for joining me tonight. Michael Diamond from Washington University, thank you so much.
It was great to be here.
Michaela Gack at the University of Chicago, thank you.
It's a pleasure.
Laura Kramer, New York State Department of Health in SUNY Albany, thank you very much.
Thank you. It was fun.
And Charlie Rice, Rockefeller University, thank you
My
so
pleasure.
much. I want to thank the meeting organizers, especially ASM President Lynn Enquist for allowing us to do this, ASV President Grant McFadden. Together they put together the meeting for tomorrow. And as I've said many times, every science meeting should have a podcast, and this is the one for our Zika meeting. And many thanks, of course, to ASM for hosting us, and to our producer, Chris Kondayan. Thank you, Chris. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWIV is viral.
Smart.
What?
Polio. Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room. Never seen anything like this.
Anybody who has access to this facility first has to go through an IR scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.